Hello everyone, good day. This is the 1 million bioinformatician movement by Nobukin Mat Limited, and this is the Genomics introductory course. My name is Asha Akintola and I will be your tutor and guide for the duration of this course. To begin this course, we'll take us through what is the meaning of bioinformatics, the structure and function of macromolecules, and then the biological information flow, which is denoted by central dogma. Now let us begin. What is bioinformatics? The simple definition of bioinformatics is the science of storing, retrieving, and analyzing large amounts of biological information. But simply put, bioinformatics is an interdisciplinary field of science. It combines several aspects of science like computer science, statistics, mathematics, and engineering to analyze and interpret biological data. The goal is to understand the information that is stored in biological components, biological entities, probably your body, in the body of animals, in your blood, everything that makes up a biological component of an individual or a species or an organism. So we want to use these different aspects of sciences coming together and then to understand, to analyze, to interpret the biological data. So there are several questions that come up that who are those who bioinformatics? Who is qualified to be a bioinformatician? Well, there are several, irrespective of which area you are, you can dabble into bioinformatics. The bioinformatics is a very wide field, just like it was said earlier, that it is an interdisciplinary area of science. You don't necessarily have to be a biology major before you can be a bioinformatician. You can be a computer science major, information engineering, mathematics, statistics, and the likes. So, and here is a Venn diagram comprising, showing us how the information, these different fields, how they are interwoven with one another. But interestingly, as a bioinformatician, you also have an element of being a statistician, a data scientist, a biologist, a programmer. So it is a very wide field that accommodates a lot of people. Now, what are some of the applications of being a bioinformatician? A lot of applications are out there to become a bioinformatician. So you can work as a researcher, you can work in the industry, you can work as an academic. So there are different areas of bioinformaticians and its applications. Now, the relationship between the molecular biology and an omics science. So molecular biology has a very strong relationship with bioinformatics, or here we call it the omics science. Genomics, transcriptomics, proteomics, and all the omics science. So in the area of molecular biology, where you're talking about DNA, that is the area we'll be talking about genomics in the area of bioinformatics. Here it answers the questions of what happens to the genes that are contained in the DNA. In the area of RNA in the molecular biology, it talks about transcriptomics, and here we find out what appears to be happening. In the area of proteomics, will tell us what makes it happen, like what makes what is happening happen. And then in the area of metabolites, where we're talking about metabolomics, that is where we'll discuss about what has happened and is happening. At this introductory stage, which is the first step in understanding this whole concept, we'll start from the genomics part, and here is the introductory part of genomics sciences. So I hope you all enjoy the journey as we take us through that. Now, the era of genomics. The era of genomics brought in the study of the DNA. So the elucidation of the central dogma, which is the information flow, we are going to talk about that later in the presentation. So it became clear that the genetic blueprint that is in the nucleotide could determine our phenotype. That is, our DNA determines how we look like. A DNA information that makes you have a blue eye, your blue eye that is shown in your phenotype is contained in your DNA. That means there is something in your DNA that tells you that you should have, or you would have, a blue eye. So those are some of the information by the as early as 1960. So, and in the recent years, there have been several scientists, there's several studies to elucidate this information. So, and this has led to this ambitious project that is called Human Genome Project, Human Genomics Project. And in this project, it was determined that the human genome has 3 billion base pairs, and we have about 20,000 genes, which is very, very low compared to the amount that was estimated before the project was executed. Now, this is an important aspect that has led to the science of bioinformatics as we have it today. Now, there are some areas in which we might be having a conflicting definition or understanding. There is genome, there is genomics, there is genetics. Now, what is a genome? A genome is the complete set of the DNA, including all its genes, while genomics is an interdisciplinary field of biology aiming to characterize and quantify all the genes in an organism and their interaction within the organism itself. Genetics on its own is the study of individual genes and their role in inheritance. So basically, genetics is focused basically on inheritance, how the genes are transferred from one individual to another, that is from the father to the son, or from the mother to the son or the daughter, or vice versa, as the case may be. So in the case of functional genomics, so it refers to the genomic annotation by using a computer to search for genes, to regulate the sequence and other specific sites from genetic sequences. So it is computer based, and that is where bioinformatics comes to play. As a form of a side note, we would know that insulin is the first type of biological entity, the first type of genes to be sequenced. Insulin, the protein of insulin, the insulin protein was the first to be sequenced. So now the science of bioinformatics has led us to annotating and searching and understanding and mapping the genes of an individual. Now, there are two basic culprits in all this information, that is the DNA and the RNA. The DNA is double-stranded, RNA is single-stranded, and by definition, DNA means a deoxyribonucleic acid, while the RNA simply means ribonucleic acid. However, there are different elements that makes up the DNA and RNA. And one of the important aspects is the nucleotide bases. So the nucleotide bases, in this aspect, we will not be dwelling much on the molecular biology side. Is what concerns us here at this stage is the nucleotide bases of the DNA. So we have the thymine, cytosine, adenine, and guanine. So they all have their own different chemical structure, while in the likes of RNA, there are 4 nucleotide bases of RNA. So uracil replaces thymine in the case of DNA. So you have uracil, cytosine, adenine, and guanine. So these two shows the structure of the DNA and RNA. The function, however, differs, and as we have noted here, the structure determines the function. Is one of the popular terms in the area of biology, whereby the structure determines the function. As you can see, that your hand, the way your hand is formed, or the way your hand is, determines how you're going to use it. So that means our structure determines the function of how we function, basically. So the DNA and RNA is not an exception. So based on their structure, they have differences and different uses and functions that they do. So we can read that up, and then we can have an idea of what they really do. Now, however, there are 5 key categories where DNA and RNA differ. One is the function, one is the sugar, one is the sugar base, the bases, the structure, and then the location. So, in the case of RNA, I would like to skip that part. We can read that up, but from the previous slide, we can see that's the structure, the function. The DNA replicates and stores genetic information. So it is the memory card, or let me say hard disk or flash drive, of all information that would happen to an individual, while the RNA converts the genetic information contained within the DNA to a format that is used to build proteins. So the protein is the later part from the DNA to RNA to protein. So we are still going to dwell more on that as the presentation continues. Now, however, there are 3 types of RNA. You have the messenger RNA, we have the transfer RNA, we have the ribosomal RNA, and each of these RNA have their function as it has been stated here. Now, the main crux, the central dogma. The central dogma is the explanation of how information flows from the DNA to RNA to protein, how the genetic information flows from these 3 stages. So when the DNA is converted to RNA, it is done through a process called transcription, and from the RNA to the protein, it is done in the process called translation. So transcription and translation encompasses the central dogma, and it's the process by which instructions in the DNA are converted into a functional product. So that means the protein is the functional product of the DNA. So now that is the message that is being passed by the central dogma. Now it also suggests that DNA contains the information needed to make all our proteins, and the RNA is the messenger that carries this information to the ribosomes. So it's in short, that is the simple explanation of the central dogma, the process, and what it's aimed to achieve. Now, there are questions that comes up that this central dogma, does it always apply to every situation? So now there is a lot of research that is focusing on the non-coding RNA. So basically it means that not all the RNA that is being transcribed from the DNA are functioning. So there are non-coding RNA, and what is their function? So research is currently focused on that too. So we are still going to dwell more on the non-coding RNA in subsequent sessions. So this is just an category of the nucleotide names of the bases. So we have the ACGT, we have the purine, and we have the pyrimidine. And this is the structures. And now we have what we call in the complementarity of a DNA. So a purine will always pair with a pyrimidine, as you can see, A, which is adenine, would always pair with thymine, and guanine will always be paired with cytosine. In our previous slide, we showed us that purine and purine category is adenine and guanine. That is adenine and guanine and the purine, and the pyrimidine is cytosine and thymine. And here we show us that cytosine and thymine, which is the pyrimidine, will be pairing with adenine, guanine and adenine respectively. So that is to show us the complementarity. Always you will be seeing this pairing. To make us understand further, we said from the previous slide that there are two strands of DNA. So in every DNA there is this pairing. So one part will be adenine, the other part will be thymine. One part will be guanine, the other part will be cytosine. So anytime you see adenine, when you look at the other part, you'll be seeing thymine. So that is what this simply means. So now, there is what we also call the palindromes in DNA sequences. That is where you're having a complementarity of a strand. So, and that is what we simply explained now. Now, we also have what we call reverse complementarity, reverse complement of a DNA sequences. Now, in the sequences given here, you can see that we have A, T, G, C, T, G, A, and the likes. But we know before that A would always be pairing with T. Now, when we take from behind this particular sequence, we can see that this A that is supposed to pair with T is starting from this end. T pairing with A, this end. G pairing with C, this end. So that is how you'll be seeing it from the end. And what we call a palindrome, it's not really a technical term that requires, palindrome. It's just like when you write dad, D-A-D. So when you read it from the beginning of the word, you have dad, of course. And when you also take the word from behind, D-A-D, you start the letter from behind. D will come first, followed by A, followed by D. Well, you also get that. That means whichever way you're having that. So that is what reverse complementarity seems to is explaining. So as you can see in the figure here, you can see the pairing. Now this is further explanation of for the case of RNA. We said uracil is replacing thymine in the case of RNA. Now, there are times in which RNA structures also fold. Why would they fold? Because we said they are single strands. Why would they fold? Well, that is the understanding we have about, that science have about RNA. RNA will also fold, and that folding will make them have the double strand, like what looks like a double strand, but in reality they are single strand. Now, to analyze, to explain further how the folding of RNA works, it's like using, when you have a sellotape, you know, one end of the sellotape is sticky, the other part is not sticky. Now, but when you leave the sellotape hanging for long, you will notice that if there is much disturbance, you will see that the sellotape will twist around itself and will stick to itself. So that is the explanation of how the double strand structure of the single strand RNA works. So it's not really, in essence, it's a double strand, but it is a single strand that has coiled around itself, and this coiling will also follow the same sequence, the same format in which the complementarity that we explained earlier on is all about. As you can see here, A is pairing with U, which U in the DNA is thymine, so A is pairing with U, C is pairing with G, and so on and so forth, as is shown here. So that is the first part of our presentation.